everyone. We're back for part 3 of the video lectures for Chapter 7 on membrane structure and function. You recall that at the end of part 2, we had sort of summarized the various means through which solutes move across membranes, passive transport, active transport, and so on. And at one point during the second video lecture, I suggested that, especially with regard to active transport, there are cases where a cell wants to set up a concentration gradient. It wants to have differences in concentrations of various ions on one side of the membrane versus the other. And the reason why a cell, well, one of the reasons why a cell wants to do that is because it can actually use that difference in concentrations, the concentration gradients, that is, to perform work. And so that's kind of what we're going to start talking about here next with this very first part of the video lecture part 3. So there's something referred to as membrane potential. Okay, membrane potential. Membrane potential is a difference in voltage on either side of a membrane. So it's the voltage difference across a cell membrane. You may not realize it, but electrical current is actually a very important part of the way that cells and organisms at a larger scale actually work. Think about our nervous system, for example, electrical nerve impulses traveling from one cell to the next. But membrane potential is the voltage difference across a membrane, and it's a force that can actually be used by a cell to perform work, okay? So how does a cell then create voltage differences across the membrane? Well, it all goes back to differences in the distribution of positive and negative ions across a membrane. So having concentration gradients of ions across a membrane is what sets up membrane potential. So up to this point, we've talked very generally about concentration gradients as a means, or a force rather, that causes ions or other solutes to move across a membrane from high concentration to low through diffusion. Well, that is certainly one thing that causes solutes to move across the membrane, simply them wanting to move down their concentration gradients from high concentration to low. But there's a second force kind of rolled in with this, okay? And the second force has to do with the voltage difference. So overall then, we have an overriding force that causes ions, other solutes, whatever, to move across a membrane. And that overriding force is referred to, or is known as, an electrochemical gradient, okay? An electrochemical gradient. This is the combined forces of concentration gradient and voltage difference, okay? The voltage difference, again, is membrane potential. So, excuse me, what we have here, then, in terms of the electrochemical gradient, is two parts. We have a chemical force, and that is simply the concentration gradient of the ion. And then we have an electrical force, okay? And that's the membrane potential. That's the difference in voltage across the membrane. So think about it this way. You have a positively charged ion in high concentration outside of a membrane and a low concentration inside. So the concentration gradient, the chemical force, causes that ion to want to move from high concentration outside toward low concentration inside. Okay, that's it, simply moving down this gradient, the concentration gradient. But it's a positively charged ion. So if there are more positively charged ions outside the membrane than inside, by default, you have a relative negative charge inside. Positive outside, negative inside. And so it's a positively charged ion. What do opposites do? They attract. And so it will tend to be attracted to the inside or negatively charged inner surface of the membrane. That's the electrical force. So both the chemical force and the electrical force will tend to draw that positively charged ion in through the membrane to the inside of the cell. Collectively, then, these two forces make up the electrochemical gradient. All right? Okay, so this electrochemical gradient then represents potential energy. Potential energy. Potential energy, again, is energy that's stored in some way that can be used to do work. An electrochemical gradient represents stored potential energy. So, cells have active transport proteins called electrogenic pumps that pump ions. We've already seen one. We saw in the last video for this chapter the sodium-potassium pump in animals. It's one of the major electrogenic pumps of animal cells. Why do we call it an electrogenic pump? Because it pumps ions, sodium and potassium ions, and maintains a concentration gradient of both. Plants don't use the sodium-potassium pump, so instead they use what's called a proton pump, a protein that pumps hydrogen ions, H+, as we've talked about in previous chapters. Okay, so whether it's the sodium-potassium pump in animal cells or a proton pump in plant cells, both are electrogenic pumps that help store energy that can be used to drive cellular work. So let's look at the proton pump. Okay, so imagine this is a plant cell. There's our membrane. Now the cell wall has not been shown just to simplify this figure, but we have the cytoplasm inside the cell and we have the extracellular fluid. Extracellular means outside the cell. So we have a proton here, and this proton pump is going to pump that proton using the energy of ATP from low concentration inside to high concentration outside. So that's why it's active transport using energy, because it's pumping from low concentration to high. Protons have a positive charge, and so as the concentration of proton goes up outside, the amount of positive charge on the outside of the membrane increases, and by default that leaves a relative negative charge on the inside of the membrane. So here we see the electrochemical gradient. We see the chemical gradient, which is simply the concentration gradient, high concentration protons versus low. And we see the chemical, or the electrical part of this, which is the positive charge outside as a result of the high concentration of positive protons and the resulting negative charge inside. That's the electrochemical gradient. So think of the positive charge out here and the high concentration of protons as water stored behind a dam. It represents potential energy that can be used to do work. All right, so let's see what kind of work. Well, we have a process that takes advantage of this electrochemical gradient. It's called cotransport. And here you see an example of this. So cotransport, prefix co, two or more things sort of working together, right? Okay, so we have a proton pump, pumping protons from low concentration inside to high concentration outside, using the energy of ATP to do that. That sets up our electrochemical gradient. Positive charge, negative charge, high concentration of protons, low concentration. So these protons now want desperately to move back down their gradient to the inside of the cell. They're moving down their concentration gradient, but also being positively charged, they're attracted to the negative charge on the inside. That's the electrical part pulling them in. So the electrochemical gradient then is wanting to pull those protons back into the interior of that cell. But being ions, they can't get through that membrane. Remember, phospholipid bilayers are essentially brick walls to most ions. They can't get through it. All right. So we have another protein here called a sucrose proton cotransporter. All right. So a plant wants to have a high concentration of sucrose inside of its cells. Okay, so imagine this is a root cell, right? Root cells don't undergo photosynthesis, so they can't produce sucrose on their own. That's the job of the leaves, which are above ground in the sunlight. But the leaves can transport that sucrose down to those cells, and those cells therefore need to get that sucrose in. But usually there's going to be a higher concentration of sucrose inside the root cells than outside. So moving sucrose in would be moving sucrose from low concentration to high. That's against the gradient, as we've talked about, right? Right, sucrose moving against its concentration gradient. Okay, so this is what plants use cotransport for. Remember, plants have built this proton concentration gradient. They built this electrochemical gradient using the proton pump, and as I said, it represents stored potential energy that can be used to do work. All right, so the plant wants to pull sucrose in, but that's going to be a case, you would think, of active transport because it's moving it from low concentration to high. But the plant wants to do this as efficiently as possible. So So it takes advantage of the fact that the proton electrochemical gradient is already there. Now the plant is setting up this electrical chemical gradient for a variety of different reasons, not just to transport sucrose, okay? So it's spending this ATP to set up this gradient for a wide variety of different reasons. It needs it for a lot of reasons, but it's there, so the plant takes advantage of it. Proton comes in, wants to move down its gradient, so it binds to a certain site in this sucrose proton cotransporter protein. Once it binds, sucrose binds to another site, and the proton flows through, kind of dragging the sucrose in against its gradient. It's almost like if you open a door in a dam and the water flowed through down its gradient, but a fish got sucked in along with it and spit out into the river on the other side, the fish would be the sucrose, right? So the sucrose is moving from low concentration to high, but you don't see this protein using any ATP to do it. What's the energy source? Anytime you're moving something from low concentration to high, it's active transport, so it has to use energy. But no ATP here, so what's the source of energy? The electrochemical gradient, the, quote, water behind the dam that I said can be used to do work. Protons flow down their gradient, and that exergonic flow, or energy-releasing flow, then drags sucrose against its gradient. So that is cotransport. It's a way to move sucrose in more efficiently because the ATP used here by the proton pump is going to be used anyway. It's used to set up this electrochemical gradient that the cell needs for a variety of different reasons. All right. Okay, so now we're going to shift gears a little bit and talk about, as the last part of this chapter, what's called bulk transport. Okay, bulk transport. So in the case of bulk transport, a cell is doing a couple of different things. It can be moving molecules, large quantities of molecules across the membrane much faster than could ever be taken care of by a transport protein or something like that. Buying in bulk, right, that kind of thing, moving something in bulk, a lot of it at one time. Or it can actually, or it can be moving a molecule that's simply too big to fit through a protein, like a large sugar molecule, let's say, or something like that. But in the case of bulk transport, the cell is going to use vesicles, okay? It's going to use vesicles to accomplish this. So we can have, as we've already talked about, small molecules in water entering or leaving a cell through the lipid bilayer or through transport proteins, as we've already said. But large molecules like big polysaccharides, proteins, that kind of thing, they're going to move across the membrane in a bulk way, through a bulk mechanism rather, using vesicles. Now, this does require energy, and there are a variety of different forms of it. Okay, the first type is referred to as exocytosis, and we've already seen an example of this when I was talking about the way that the ER and the Golgi complex work. I said the ER transports proteins down to the Golgi. Golgi processes those, ships them off from the other side, and in a vesicle, and those vesicles then fuse with the cell membrane and dump those contents off to the outside. That's exocytosis. Exo, exterior, moving something to the exterior of the cell, okay? So here, transport vesicles migrate to the membrane from the inside, they fuse with it, and release their contents outside the cell. This is the way a lot of secretory cells produce products that end up in your bloodstream, like hormones being released by a gland or something like that. Insulin being produced by your pancreas, for example. All that insulin ends up in your bloodstream through exocytosis. So let me play this little animation here to explain this.
Exocytosis is the movement of intracellular vesicles to the plasma membrane, where they fuse with the membrane and release their contents into the surrounding fluid. This process occurs predominantly in secretory cells, such as mucus-producing cells or pancreatic cells, that secrete enzymes into the digestive tract.
Okay, so extracellular processes like this are moving things from inside to out. Okay, that's exocytosis. It's fairly straightforward. The reverse of that, taking something in from the outside, is called endocytosis. Endocytosis. Endo, interior, taking something in. So here the cell takes in large macromolecules by forming vesicles from the plasma membrane, and they pinch inward. It's a process called invagination. Okay, we'll see that in a moment. Endocytosis is basically the reverse of exocytosis, but there are different proteins involved here. Okay, first of all, we have three different categories of this as well. We have, first of all, what's called phagocytosis. This is also referred to as cellular eating. So this is when a cell takes in a particle of some form, something solid. It could be a bacterial cell, or it could be just a particle of detritus or organic material, whatever. When a cell sort of gulps in or drinks in fluid, that's a type of endocytosis referred to as pinocytosis, also called cellular drinking, taking in fluid and whatever might be dissolved in it. And third, we have a very specialized form of this called receptor-mediated endocytosis. This is where you have proteins in the cell membrane that are sensing the presence of a very specific kind of molecule outside the membrane. When the molecule binds to those receptor proteins, the membrane invaginates, pulls them in, gulps them in a vesicle, and captures those molecules, those target molecules specifically. It's a much more specific and targeted type of endocytosis. Okay, so let's take a look at these examples. So here we have phagocytosis. So you have a particle of food of some sort that bumps into a cell membrane. Maybe this is an amoeba cell. And that membrane will invaginate or bow inward and pull that particle of food in, and pinch off on the other side of it, and trap it as now a food vacuole. A lysosome will then maybe fuse with that, dump off its digestive enzymes, and digest that particle of food. That's the way amoebas, single-celled organisms, will eat, for example. Pinocytosis. So here the cell membrane invaginates again, pulling fluid and whatever might be dissolved in that along with it. Cellular drinking, in other words. Okay. In receptor-mediated endocytosis, now you see we have these red V-shaped proteins. We have these purple molecules here. By the way, those purple molecules are called the ligands. Okay, ligands. They're the target molecule. They will bind to that receptor protein, and that way the membrane of the cell recognizes that the ligand molecules are there. It imaginates, bows inward, and pulls them in and traps them. This is called a coated vesicle now. Okay, so again, in this case, you're targeting, or the cell is targeting, a very specific molecule, and not just whatever happens to be there, as is the case with pinocytosis or really phagocytosis as well. All right, let's look at another video.
In endocytosis, membranes invaginate or pinch in to form a vesicle, moving the enclosed materials inside the cell. This process can take different forms, each involving its own specific cell machinery. In phagocytosis, or cell eating, the cell engulfs debris, bacteria, or other sizable objects. Phagocytosis occurs in specialized cells called phagocytes, which include macrophages, neutrophils, and other white blood cells. Invagination produces a vacuole, which usually fuses with one or more lysosomes containing hydrolytic enzymes. Materials in the vacuole are broken down by these enzymes and degraded.
Okay, another video.
In endocytosis,
Oops, sorry, same one. Another video.
In pinocytosis, or cell drinking, the cell engulfs extracellular fluid, including molecules such as sugars and proteins. These materials enter the cell inside a vesicle. Epithelial cells in capillaries use pinocytosis to engulf the liquid portion of blood at the capillary surface. The resulting vesicles travel across the capillary cells and release their contents to surrounding tissues, while blood cells remain in the blood.
Okay, and lastly, we have...
In pinocytosis, or cell drinking,
Lastly, we have a video for receptor-mediated endocytosis.
unlike phagocytosis and pinocytosis, receptor-mediated endocytosis is very specific. It is triggered when membrane receptors bind to specific external molecules, such as protein cholesterol complexes or proteins bound to iron. Membrane vesicles pinch off, and the external protein and its cargo are brought into the cell.
Okay, So we've now then come to the end of the chapter. We've gone through the various ways that substances enter and leave cells. We talked about passive transport. We talked about active transport. Back in passive, we singled out osmosis in particular. We've talked about bulk transport, co-transport, all that sort of thing. It's a lot to keep straight. But at the end of this chapter here, we have a BioFlix animation. I don't think I've shown one of these yet, but these are some fairly high-end, very well done animations, good graphics and that sort of thing that show these processes. I've chosen to add one here at the very end of this because this one sort of summarizes the various ways that we've talked about that things can move across membranes. So I'm going to play this, and that will be the end of the video lectures for Chapter 7.
We get all our energy and organic molecules from food. Before we can use the molecules we eat, they have to enter our cells, starting with the cells lining the small intestine. Let's zoom in to the surface of a cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can dissolve in the lipid bilayer. Notice how they move down their concentration gradient, from where they are more concentrated to where they are less concentrated. This is an example of diffusion. Diffusion is a form of passive transport. It does not require energy from the cell. Most molecules can't cross the lipid bilayer. Here, the sugar fructose moves into intestinal cells by facilitated diffusion, moving down its concentration gradient through a transport protein. Facilitated diffusion doesn't require energy from the cell, so it's also a form of passive transport. Water crosses the plasma membrane by facilitated diffusion, or by diffusing across the lipid bilayer directly. The diffusion of water across a membrane is called osmosis. The sodium-potassium pump moves ions against their concentration gradient, from where they are less concentrated to where they are more concentrated. This requires energy from the cell and is known as active transport. Energy from ATP is used to move sodium ions out of the cell and potassium ions in. Another type of active transport is co-transport. Here, both sodium ions and glucose move into the cell through a co-transporter protein. Sodium ions move down the concentration gradient created by the sodium-potassium pump, and glucose moves against its concentration gradient. Now let's move to the other side of our intestinal cell. Materials can be exported in vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, the plasma membrane pinches in, forming a vesicle that contains material from outside the cell. On this side of the cell, we can also see oxygen and carbon dioxide diffusing across the lipid bilayer. Cells use all these processes to get what we need.
Okay, so that's it then for Chapter 7.